লাইভ করে দিচ্ছে না
ব্যাটারি
হ্যাঁ এই যে
অন করে দাও, অন করে দাও। ওই পাশে।
এই পাশেরটা আচ্ছা
হ্যাঁ, যাচ্ছে। চলো, একটু অন করো, অফ করলো তো।
অন করে আসলে তো বলতে লাইভ সঙ্গে চলে গেছে
আচ্ছা।
Laptop camera to the slides. So the people who are using YouTube,
this is how it is. This is fire and then the camera
যাচ্ছে
so it's just showing the
দেখা যাচ্ছে
screen.
দেখা যাচ্ছে ঠিক আছে ঠিক আছে
basically this is how it looks in a meeting. Sharing the work.
ঠিক আছে
Yeah, so the whatever the software is just capturing the laptop screen. It's not actually capturing this screen.
No. Yeah, so like how
আমি তো ওদের পরে কথা এবার লাইট করে দাও
লাইভ হয়ে গেছে স্যার
আচ্ছা ঠিক আছে
এই উপরের এটাকে কি কাটানো যাবে কোনো ভাবে নাকি থাকবে
ওই উপরের প্যান্টটা স্টপ প্রেজেন্টিং
থাকলে কোনো অসুবিধা নেই
থাকবে
মানে যদি কাটানো না থাকে ঠিক আছে
সরানো যাবে ওটা হাইড করে যাচ্ছি সেটা এটা হাইড করে দিতে চমৎকার
থ্যাংক ইউ না না ঠিক আছে
Okay, let's start. Oh, there's my YouTube, right? All right. So am I audible over the microphone? The microphone? Yes. Okay. Okay, it's my pleasure to introduce Kushal here. My relationship with Kushal goes back a long way when he was a final year BStat student at ISI. Uh, After that, he has gone to the University of Chicago, worked with Matthew Stevens for his PhD. And then he went to Harvard, where he currently is at the Harvard T.H. Chan School of Public Health. And he is a postdoc working with Alcus Price. So he has worked with stellar individuals and he has done wonderful, wonderful work, some of which I think we will have the pleasure of hearing. If we can press him hard enough, then maybe he is here for a while. So maybe we can get him multiple times into the institute and learn multiple things from him. Okay.
Give us seminar a week.
Let's get a seminar series from him. Yeah.
Every week, one seminar.
Once. All right. Without much ado, Kushal, it's all yours.
Thank you so much, Anurabha, and thanks everyone for coming. So today I will talk about three broad topics that I have been working on during my postdoc, and I hope to continue working on those going forward as well. And the idea kind of centering all those, or basically, if there is one particular larger topic that I'm interested in overall, is to identify the functional mechanisms of GWAS variants. So just to give a little bit of a background, in the past nearly 17, 18 years of genome-wide association studies, we have been able to identify nearly 100,000 GWAS hits spanning thousands of traits as of 2021. Despite looking at many traits and diseases and performing these GWASs, there have been certain challenges in translating these GWAS discoveries into therapeutics. Two primary challenges are, number one, most of these complex diseases and traits are highly polygenic in nature, meaning that there are many genes that are implicated by these variants rather than one or two genes, unlike some Mendelian disorders. And more than 90% of these GWAS hits have been found in the non-coding regions of the genome, which makes this all the more challenging because for a long period of time, very little was known about the functional activities in the non-coding regions of the genome. So there are, however, some positive signs recently. One from this 2015 Nature Genetics paper showing that drug targets with genetic or GWAS support have been found to be twice as likely to be approved. And there is one really interesting paper that just came out last week, where they actually looked at the 50 FDA-approved drugs in 2021, and they found that 33 of those 50 drugs had genetic support. And by genetic support, meaning that there were genes that were implicated either directly by GWAS or there were proteins that were physically interacting with those genes. And if you just look at this particular graph at the bottom, you will also see that if we look at the common disease row, so all the columns are these 50 drugs. There are 50 columns here, but you will see that for the common disease, actually we have a large fraction. So a large fraction of these 33 approved drugs are actually implicated by common diseases rather than somatic mutations or rare Mendelian diseases. So this kind of highlights the need to focus more on the common disease mechanisms driving a lot of the disease and traits that we see around us. And the hope here is that if we can try to understand the functional mechanisms of how these variants are affecting the disease, then maybe we can do an even better job at generating better drug targets. And maybe this number 33 out of 50 could even be higher if we can kind of pinpoint those genes better. So what I mean here is that while we know that what are the variants that are associated with disease, we also need to know what is the regulatory activity at those variants, because most of these variants are kind of non-coding in nature. So they must be somehow affecting genes. So we would like to know what sort of regulatory signal do we see at these variants. Then we also want to know how these regulatory activity at these variants are translating into changes in the expression of genes that may be linked to these variants. And that's not all. We also need to then understand how each gene is interacting with other genes, because a disease mechanism is, because this is a complex disease, so there are many genes. So one of the ways in how in which all these different genes are probably associated with the disease is through gene-gene interactions. And to understand those gene-gene interactions better, and also to understand how these genes interact with each other in specific cell types and tissues, because in many cases, the traits and diseases are related to certain tissues or certain cell types. And to identify what are those disease critical cell types is also important. It is also important to identify that for doing functional follow-up experiments. Like if you want to do some CRISPR experiments trying to perturb a particular region or can do some variant level base editing, you also need to know like what sort of cell lines or what sort of cell types to focus on. And so it's important to understand the underlying cell types or cellular states that are important for the disease architecture. So to this end, I will talk about 3 projects. The first one is going to be about prioritizing variants for disease critical function. And then I will talk about how to identify which cell types are important for disease. And third, I will talk about identifying genes and gene pathways that are important for disease. So in order to identify which variants are actually important for disease, and this is from the point of view that when we say that we want to prioritize a variant for function, we basically want to understand whether that variant is important for disease, but at the same time, whether we see a lot of evidence from functional genomic data that tells us that this variant is going to be an important variant. So one of the things that we do here is we have aggregated a lot of base pair resolution functional data. So for example, you can look at allelic imbalance of the DHS or TF chip activity. So it basically means that if you have a particular affinity of a transcription factor binding at a particular site, then how much does the change of the variant from the reference allele to the alternate allele, how much does that change the transcription factor binding affinity? The same thing you can do for DHS or chromatin accessibility as well. So by doing that, you can actually tell, like at a scale of a single variant, what's the effect of the allele change on the regulatory activity at that region. You can also look at QTLs. We're all familiar with expression QTLs, like the variants that are causing changes in the expression of a gene. But we can also look at chromatin accessible QTLs or other types of QTLs like methylation QTLs as well. There is also, we have been looking at some MPRA reporter assay-based assays where we synthetically alter the variant from the reference to the alternate, and then we see what's the effect of that on the expression of a fluorescent gene that is downstream from that particular variant. So it basically highlights the overall enhancer or change in enhancer activity at the resolution of a particular variant. The fourth component that we use is kind of sequential model based, and this is using deep learning models. I will not go very deep into the deep learning modeling framework about how we are obtaining these variant level scores. But the reason why we are excited about these deep learning models is that all these experimental assays, they would cover a fraction of the overall variance that we see in the genome, and primarily would be driven by some of the common variation. But using the deep learning models, you can actually find a prediction of regulatory activity at a particular, the resolution of a particular variant for any variant, whether it is a common variant or a rare variant or a somatic mutation, you would be able to do that. And I will show in the next slide how one can do this. The idea is to then use all these experimental base pair resolution functional regulatory effects, and as well as the deep learning computational based effects, and aggregate all of these things together to come up with one single score of prioritizing variance for function. And we call this a consensus variant to function score, or CV2F. A variant which has a high CV2F. So this CV2F is a probabilistic score, and I will show how we are generating this. If a variant has a higher CV2F, that basically means that it has a lot of evidence for function from multiple sources, be it experimental or from computational models. But you can then also dig deep into those variants, and you can see like what are the cell types in which the allelic, if it is implicated by allelic imbalance experiment, you can check what are the cell types for which we see the allelic imbalance, or for which or what are the assays, like whether it is picked up by allelic imbalance or for QTLs, or like or both of those assays. So you can also see all of that information. So the consensus variant to function is more of a way of cataloging variance based on function, and kind of having a hierarchical way of looking into the overall function, and then digging deeper into finding the exact mechanism of that function based on cell types and assays. So before we talk about how we are generating this CV2F score, I'll just briefly mention how we are generating these variant or allelic level effects from these deep learning models. So the way that it is done is we have a lot of ENCODE and roadmap epigenomics data for different chromatin marks. Like we have DNAs, we have histone marks, we also have transcription factor binding assays for throughout the genome. We can actually see what are the regions where we, or what are the peaks for each of these particular assays, and which regions are implicated by each of these assays. And now what you can do is you can take sequences from across the genome of a particular size. Either you take like 1,000 base pair sequence, or you can even take a longer one. And now for this particular sequence, based on where it is coming from the DNA, you know what was the DNase or H3K4-ME1 or ME3 signal at that particular location for the different cell types. The idea is to train this deep learning model, where as input, you are inputting all these different sequences from across the genome. And as output, you are telling the model what are the DNAs and H3K4-ME1, ME3, and DF binding features that you are seeing at the location of this sequence. The hope is that once you have trained the model well enough, now you can look at a particular SNP, and you can look at the contiguous sequence around that particular SNP for both the reference allele and the alternate allele. For both of these sequences, the reference sequence and alternate sequence, you will get two predictions for each feature. Let's say you're interested in DNAs in T cells, you will get a PF and the QF for this particular feature. And then you can look at the magnitude of the difference between the two, which we are calling as the allelic V2F effect. And just based on how it is this score is obtained, you can actually tell that you can do this for any variant or any mutation in the genome. You can actually look at the sequence, and you can look at the differences in the predictions for that particular mutation. Now, once we have these deep learning based variant effects, and we also have other variant effects from these experimental assays, we can use them as features and try to see whether these features or what combination of these features are predictive of GWAS fine-mapped causal SNPs. So what we did was we looked at strongly fine-mapped causal variants from multiple GWAS traits, like around 90 different GWAS traits from UK Biobank. And we picked as controls a set of SNPs for which we see very low posterior inclusion probability or very low evidence of causality across all the traits. And then it is about classifying these set of fine-mapped causal SNPs from this set of non-causal variants using all these experimental assay and deep learning-based variant to function features. Now, the way we are generating the CV2F score is by performing this classification task basically 22 times. So let's say that I want to generate a CV2F score for all the chromosome 1 SNPs. What I do is I perform this classification experiment for all the variants from in chromosome 2 to 22. And then the training model that I get, I use that to predict on the SNPs from chromosome 1. And that is going to give me a prediction of how likely this particular SNP is, how likely to be causal this particular SNP is based on the functional annotations at that SNP. And now you can do the same thing 22 times for each of the 22 chromosomes. And that will give you the CV2F probabilistic score. Now, how can we use these CV2F scores? So one example is here. So for lymphocyte count GWAS trait in UK Biobank, there is this one locus in chromosome 8, which is a 12 kb long locus. And all the variants in this locus have very strong GWAS association p-values. All of them are significant GWAS hits for lymphocyte count. And we wanted to know like which SNP out of these are causal SNPs. But it turned out that it is very difficult to pick the exact causal variance in this case, just purely based on GWAS data. I mean, one thing that you can do is perform GWAS fine mapping to identify the likely causality of this variance. But the problem here is that the LD in this region is really high. So the average D prime is close to 1, which makes any statistical fine mapping approaches kind of invalid in this context, which is why you see this fine map PIP scores to be uniformly low throughout this region. So now what we do is we overlay the CV2F scores on top of all these variants. And you can see that there is this 3 variants in the middle, which actually have much higher CV2F scores compared to the other variants that are there in this particular region. And so now, if one has to perform functional experiments, probably trying to see whether this which variant in this region is probably causal, they probably can just focus or start with these 3 variants instead of looking at this entire 12 kb long region. One question that you can also ask, like, OK, we pick this particular variant or this set of variants in the middle, like why are these variants, why do they have a high CV2F score? So it turns out that at least for this one in the middle, we actually see evidence of functional base pair resolution effect from both DNS allelic imbalance, the TF-chip allelic imbalance, and the fine-mapped EQTLs in GTEx, and also from deep learning models. So it basically means that there are 4 different layers of evidence, which are all orthogonal assays, basically, that are implicating this particular variant for function. And so, so it definitely means that it is a good candidate variant to probably try CRISPR-I or functional follow-up experiments on. The other thing that we can do with this CV2F score is we can compare it with the general pathogenicity scores that we typically have across the variants. So here we are comparing it with the CAT score, which is a popular pathogenicity score that is typically used. And we wanted to see like whether the CV2F variants actually explain higher disease heritability signal across traits compared to the CAT score. So we performed the stratified LD score regression model on the variants that are annotated by CV2F and then compared them with the variants that are annotated by CAD. And we performed a meta-analysis either across all traits or brain traits or blood traits. And we saw that meta-analyzed across all traits, the CV2F was almost 4 times more disease informative than the CAT score. And it was even higher when we looked at meta-analysis across the blood-related traits. So far we talked about how to prioritize variance for function and to use that for understanding the disease architecture at base pair resolution better. Now for the next part, I will talk about how to identify cell types in a tissue that are likely critical for a particular disease. So what we are mainly interested here is that, let's say that I'm interested in Alzheimer's. I want to know what are the cell types, or maybe if we are interested in immune cell types, like we want to know what are the immune cell types that are important for Alzheimer's. And let's say that we find that monocytes are an important cell type for Alzheimer's. We then want to know what are the genes that are driving this cell type and disease association for Alzheimer's. So now the question is that how do we get at this cell type resolution? And one of the ways in which we can define a cell type is by using a program of genes or a set of genes. And here we do that by looking at genes that are specifically expressed in a particular cell type compared to the other cell types in a tissue. So for example, let's say that we have an immune PBMC data, and we can perform the clustering of that data and look at this is the UMAP representation of this, and you can see that they cluster by the different cell types. So now what we can do is we can look at genes that, for example, this MS4A1 gene, it is actually much more highly enriched in the B cells compared to any other cell types, or in or average across any other cell types. So the MS4A1 is going to be present in the cell type program for the B cells, but it is not going to be a part of the cell type program for any of the other cell types. Similarly, we can do it for the LYZ, for example, will be in the cell type program for monocytes, IL-32 in the T cells, etc. So now the question is that, so what we did was we looked at different tissues from across the human body, and we looked at these cell type level gene programs from these tissues. And now the key element in this method is to connect SNPs to these particular genes. So at the level of a cell types, or the thing that mainly distinguishes one cell type from the other is differences in regulatory activity between the different cell types. And the large part of that is driven by enhancers, which are usually like maybe intronic or maybe intergenic, but usually more distal to the gene than maybe promoter or coding based architecture. So what we do here is we look at some predicted enhancer gene links. And we use two methods here, activity by contact method and the roadmap enhancer gene linking method. So these are two methods that can actually predict based on a histone mark and DNA's combination, which regions are enhancers, and then they use loop information from the enhancer, like the 3D chromatin interaction models, to identify these enhancer promoter loops. So we use that information to figure out what are the SNPs that are linked to genes that are part of these cell type gene programs. So once we have that, we can actually generate an annotation of SNPs. So let's say that I have a particular gene set, and I am now combining the SNPs that are linked to the genes in this gene set. And so the genes that are part of the gene set are marked in red. Now any SNP that is annotated, that is linked to a gene in the gene set, will get an annotation of 1. And if that SNP is not linked to any gene, or if it is linked to a gene that is not part of the gene set, then that SNP gets an annotation of 0. So in this way, you can create a SNP level annotation corresponding to a particular cell type and a cell type gene program. Next, you can actually assess the disease heritability enrichments for this particular annotation that you generate. And here we are primarily looking at this enrichment-based score, this E-score, which is a function of the overall heritability enrichments that we see in this annotation. One of the interesting results that we found was that when we applied this particular method to these cell type programs from the different immune cell types, and then assess their disease signal for a number of blood cell traits, we actually picked up the related blood cell trait and cell type pairs pretty well. For example, the B cells and T cells were more enriched for the lymphocyte percentage than the RBC count and RBC distribution with traits were more enriched for erythroid, platelet counts, megakaryocytes, etc. So it did seem like at the cell type resolution, we are able to pick up this cell type specific architecture for this cell type related traits pretty well. Looked at a sensitivity specificity index, kind of looking at the average difference in disease signal between the boxed pairs of the cell types and traits which are related cell type trait pairs versus the other pairs. And we observed that using this roadmap, this combination of activity by contact and roadmap enhancer gene links, we can actually get a much better sensitivity and specificity, which will be completely missed if you had looked at a 100 kb window for linking SNPs to the genes in the program. Now you can expand to look at different autoimmune disease traits. And one of the things that we find, for example, is that for B cells, T cells, dendritic cells, we see a lot more signal across the different autoimmune diseases. But on the other hand, if you look at the erythroid, we do not see much signal across the diseases, which effectively says that we are basically picking up immune cell types for immune diseases, which is a nice validation. But at the same time, we also see some interesting patterns, like for Alzheimer's, we see signals in the dendritic cells and monocytes, but not much in the B and T cells or the erythroid. For ulcerative colitis, we see higher signal in the B cells compared to the T cells. And for the celiac, we see higher signal for T cells compared to B cells. We also performed a similar approach for brain single cell RNA-seq data. And we looked at GABAergic neurons, glutamatergic neurons, and non-neuronal cell types and evaluated them for different brain-related traits. One thing that we observed that the non-neuronal cell types were not very informative for any of these psychiatric neurological diseases that we looked at. And also among these different brain-related traits, we observed a clear distinction of some of the traits being more GABAergic in nature, like the BMI and major depressive disorder, and then some other traits being more glutamatergic in nature, like schizophrenia, intelligence, etc. So now the question is that now we have a disease critical cell type. So how do we go from the disease critical cell type to identifying the genes that are driving the disease critical cell type? So what we do here is for every enriched cell type and disease association, we'll look at the genes that have a very high program grade, like greater than 0.8 in that particular cell type gene program. And then we order those genes based on magma gene level association score. And we find that that gives a pretty nice results. So for example, if we look at heart single cell RNA-seq data and evaluate them for heart related traits, one of the associations that gets picked up are atrial cardiomyocytes and atrial fibrillation. And if we look at the top 3 genes from the association, we find that all of these top three genes are ion channel genes, and ion channel pathway is known to be an important pathway for atrial fibrillation. Now, finally, outside of prioritizing variants and prioritizing what cell types, yes. Yes.
Are you using a specific kind of disease or what are you doing?
Yeah.
I mean, I didn't understand this part.
Yeah, so originally for the gene programs, we are just using this differential expression analysis here, based on which genes are more enriched in expression in that particular cell type compared to the other cell types. So now, once we find it, yeah, so these are from the brain single cell RNA-seq data.
For specific to the brain or entire brain?
No, so this was for the entire brain. For example, in this particular, so this was based on the, so this data set here was actually from the bone marrow. That were related to the blood cell traits and autoimmune diseases. This one was from the brain that we used to connect to the different brain-related traits. And this one is actually from the heart single-cell RNA-seq data. Here, like the programs were all based on that single-cell RNA-seq data using the differences in expression. But then we are prioritizing the genes in these programs based on this magma gene level association scores, which is basically just looking at the overall density of GWAS peaks around the genes that are part of the gene program.
Sorry to have
Yeah.
interjected. Maybe I can take it up later. Go ahead.
No, yeah, please ask.
So what I was asking is
Yeah.
that you already, since you have formed the clusters on the basis of the data,
Yeah.
you already know which cluster is expressing which gene more.
Yeah.
You already know that.
Yeah.
Now you're identifying that cluster with a particular disease or disease phenotype, like atrial fibrillation or whatever.
Yeah.
That's what you're trying to do.
Yeah. So the cluster is obtained purely based on the gene expression data, and we are now identifying whether that cluster is important for disease or not. So this dot plot here is basically just doing that.
That information comes from where?
This heritability model that we are.
From previous literature.
Yeah, so basically what we are doing is that this cluster of genes that we are getting, we are converting that into a set of annotations. So basically we are looking at SNPs that are in the enhancers for those genes, and then we're looking at heritability signal for that. And based on that, we are getting this enrichment signal. And then going from that, if we have to say that, okay, what are the genes that are probably most disease informative in this particular cluster trait association signal, maybe you can use some gene level association scores to kind of prioritize genes.
So if I had GWAS data on each of these phenotypes,
Yes.
and if I have identified genes that are associated with these clusters,
Yeah.
and if those GWAS sets fall in this cluster, I'm done, right?
Yes, yeah. And so the main thing in the middle is basically identifying this cluster, which is actually important for GWAS, because that it kind of reflects a coordinated process through which these different genes are probably affecting the disease. Yes. And to that point, the next work that I'll talk about is also very important, because effectively at the end of the day, we probably want to identify what are the genes that are basically important for a particular disease, irrespective of the variants and the cell types. So for this, there are two main aspects that we have to use. One is that we want to prioritize genes for which we see a lot of GWAS signal around those genes. And at the same time, we also have to take into account the fact that in many cases, a gene might be important for disease, but it may not have a lot of GWAS peak density around it, but it may be interacting either functionally or physically with other genes, which actually has a GWAS signal around it. So that basically means that we need to take into account these gene-gene interactions and also identify how these genes are co-regulated in a particular cell type in different tissues. So to this end, we use two previous approaches. The one is completely GWAS-centric approach to gene prioritization, which is called MAGMA, where we aggregate GWAS SNP level summary data for all the SNPs in the gene body. Basically, just taking the average of the minus log p values for all the variants in the gene body, or you can also take a gene window. The other strategy, which is from a recent work from Hilary Finucane's group, where we look at genes that are not just implicated by MAGMA, but these are genes that are probably correlated in some way with the genes that are GWAS implicated genes from MAGMA. And this is based on a number of gene level features, like using single cell transcriptomic data or protein-protein interaction networks, or the pathway data that we are trying to figure out which genes are interacting with which genes, and then using that into a linear model and trying to kind of figure out how these different features can inform other genes that are not directly MAGMA or directly GWAS implicated genes. One approach that we kind of came up with, and which kind of worked pretty well, at least in terms of identifying Mendelian genes and drug target genes, was to look at genes that are actually interacting with the MAGMA genes by performing a random walk with restart algorithm on the entire graph, the string PPI network graph that we have, or we also looked at the co-expression gene co-expression networks from this co-express DB. So we looked at the both of these graphs, and then this random walk with restart, what it does is that you start from one node, and here the node is the MAGMA implicated gene, and you basically travel through the graph, but you also have a probability of coming back to the starting point with probability 0.5. That's what we took at every step. So based on that, you can see the amount of traversal that you were making at each node in the graph. And so basically what happens is that if there is a particular gene that is not implicated by magma, but maybe interacting with one or more magma genes, then that gets traversed more number of times, which is why it gets a higher priority score overall for the disease. So the final score that we came up with was this polygene, which we call as polygene score, and that actually gives an equal weightage to both the POPs method and to this polynet method. And so now so based on this polygene method, what we can do is for any trait, we can actually rank all the genes that are there for that particular trait, or we also get a score, basically, which tells us the relative importance of one gene versus the others. You can also use that to score a program of genes or a set of genes, which is quite a simple procedure here. You just take the ratio of the average polygene gene level scores for all the genes that are part of a gene set or a gene program, and then divide that by the average that you get across all the genes. So it's kind of like an enrichment based score that you are getting for the genes in the gene program. Now, one thing that we did was we wanted to assess how well this polygene method is doing compared to some of the other methods, like the magma method or the POPs method. And to that end, what we developed was looking at this program scores for a number of benchmarking gene sets and a number of traits. So the benchmarking gene set could be either a non-trait specific gene set. So for example, like essential genes as evaluated by CRISPR experiments in humans and mice. And then you can use all the traits that you have at your disposal and try to look at the overall polygene program score and compare that with program scores from other methods like magma and POPs. The other thing that you can do is look at benchmarking gene sets that are specific to a category of diseases. Like, for example, you can look at immune-related drug targets. And then you can, as a set of traits, you can look at all the autoimmune disease traits or blood cell traits for that matter. And you would expect that the polygene program score would perform better than the other methods if it is indeed a good approach. So the first thing that we did was we took a collection of gene sets that are known to be functional, but for which we do not have a particular disease or trait that we can link to that particular gene set very clearly. So we performed the polygene program score as well as magma program score and POPs program scores for these gene sets with respect to all the traits and diseases that we had, like 120 complex diseases and traits. And we observed that for all these different approaches, especially with respect to the human essential genes based on some CRISPR experiments, the polygene program scores perform better than the magma and the POPs related approaches. The other thing that we did, and this was with respect to gene sets that are specific to particular traits or particular category of traits, like immune-related drug targets or Mendelian genes that are related to blood cell traits or Mendelian genes that are related to immune dysregulation, and then link them with a candidate set of traits that are related to this particular process. For example, for immune drug targets, we can use immune diseases. For psychiatric Mendelian genes, we can use psychiatric diseases like schizophrenia, and for the BRCA genes, we use cancer-based like complex traits like breast cancer, prostate cancer, etc. And then we compared the polygene program scores for these combinations with that of magma and POPs, and we observed the polygene scores to be nearly 3 times higher compared to magma, and also 1.3 times higher compared to the POPs based scores. So these kind of highlights that polygene is able to pick up drug targets and Mendelian genes for the traits better compared to the competing methods. So in summary, we proposed basically talked about three different things, but they're kind of interconnected with each other in the sense that we are basically trying to understand the functional mechanism at the level of variants, genes, and cell types for these diseases. So first we looked at a high resolution variant prioritization score called the consensus variant to function, and it is mainly the main goal here is to identify the causality of the variants, either implicated by GWAS or even maybe not implicated by GWAS, but maybe the ones that could be functionally important. And the next, we looked at this SC linker approach where we assessed heritability signal in different cell types using primarily the main driving component here was this enhancer gene linking strategy, kind of highlighting the enhancer specific architecture for the cell types. And finally, we looked at a method to prioritize genes for disease based on both GWAS data as well as single cell transcriptomic, then pathway data, PPI interactions, and co-expression data as well. And we showed that that performs better than other methods in predicting the drug targets. For my future directions, what I'm interested in is to expand this CV2F scores that I spoke about. So the CV2F score currently is a tissue agnostic score, and so it gives an overall sense of function for a particular variant, but it does not tell us whether this particular variant is critical for function in one particular cell type and not much for the other cell type. So what we would like to do is to come up with tissue and cell type specific CV2F scores, and the hope is that if we can identify these cell type and tissue specific CV2F scores well, we can use that to generate tissue and cell type specific polygenic risk score models. So of course, like for anyone working on common diseases, one of the goals is to generate better risk score predictions. And it turns out that the risk score predictions are often not very informative, or we do see some positive signs with some of the PRS models recently, especially for cardiovascular traits, but for many other traits, the PRS models have not been very effective. So the idea would be that maybe if we can restrict them to the tissue and cell type specific scores, then maybe we can if we can inform those PRS models using this cell type and tissue-specific functional genomic data, we can probably do a better job at the risk prediction. At the same time, one of the things that we are interested in is also subphenotyping of different phenotypes. So we want to know, for example, what amount of the risk score for Alzheimer's is probably driven by microglia. And we can actually get an assessment of that based on using this tissue and cell type specific PRS models. The other thing that I'm interested in is to develop the polygene framework more by also integrating both common and rare variant data, because right now we are only using the GWAS, but we're planning to use GWAS and WES data from UK Biobank, as well as maybe other coding and non-coding information, maybe these enhancer gene links or other types of linking methods to kind of prioritize genes based on non-coding data as well. And finally, one of the things that we have kind of neglected in this entire presentation was any variant that are not common and low frequency variants. So, but we feel that once we understand the functional genomic architecture better, the resolution of cell types and tissues, we can use it not just to prioritize common variants, but also, and this is something we're still kind of struggling a lot with, is to understand non-coding driver somatic mutations for cancers and separate them from the passenger mutations. And we believe that with so much of this functional genomic data that we have at our disposal, and maybe also right now we're planning to look at a lot of this cancer cell or tumor microenvironment based genomic data. So maybe all those information can be useful to prioritize these non-coding driver somatic mutations, which because a lot of the cancer related works are kind of focusing more on the coding region. So it's probably interesting to see like how much the non-coding mutations can be important for that. So that's all that I had to present. I'll just thank my advisor, Alkes Price, and many other PIs and collaborators who were part of all these works, and also the ENCODE and the IGVF consortium who have helped me a lot with the data sets for the variant level functional architecture stuff. Thanks so much, everyone. I'll take questions. Yeah.
Very nice presentation.
Thank
so I have two questions. First question is that when you showed these the D prime score 0.99, that slide, you have mentioned that we need to look at both the analytic imbalance assay data with deep learning, or
Yeah, so basically what we did here was look at both of these.
this slide. Yes.
Yeah. So here we this CV2F score is using both experimental effects as well as the deep learning based effects together. And what we then do is that once we see that this particular SNP has been picked up, we can actually go into the feature set to see like, okay, what are the what was the like whether it was implicated by the DNA allelic imbalance in some cell types or TF allelic imbalance, or based on the deep learning models, does it have a high score based on that?
But then your deep learning method was based on these analytic imbalance data, right? Or
No, so the deep learning method is actually not based on allelic imbalance. So the deep learning method actually uses the chromatin data and the TF binding data, but that is purely based on the sequence data that is from the reference based sequence. Now, what happens is that they actually generate a predicted allelic imbalance, which is in this particular step. So here, when they compute this allelic V2F effect, this effect is a predicted allelic imbalance kind of an effect for each of these DNS features or histoma features. But the training of the deep learning model is not on the allelic imbalance data. So the training happens only on the non-allelic based, just the peak data sets.
Experimental validation along with the deep learning, and which is basically the predictive one.
Yeah, so the deep learning model is kind of like a prediction of the allelic imbalance, and that prediction you can get for all the variants genome-wide, whereas the experimental validation probably you will get for like 15% of the variation overall.
The other question I'm having, just two slides.
Yeah.
So here, isn't it that you are looking at the topologically associated?
Yeah, so what happens is that you can actually look at a TAD, and you can look at all the SNPs that are there in the TAD and then that are near the gene. One of the things that we have seen that the TADs are usually a little bigger compared to the resolution at which we get these loop informations of the enhancer and the promoters. So, I mean, it is, however, true that these enhancer promoter interactions mainly occur within a particular TAD. So, so yeah, it is kind of a TAD-based method, but I mean, it is also a little more granular than just looking at the same TAD and looking at the variants that are there.
The last question I have is that POPs and the poly. You are considering the PPI, string PPI, the polynet, and then the POPs, you have a different PPI data?
No, it's the same PPI. Yeah.
Is it like, so how do you, I mean.
Yeah, it's like the same information that, yeah. Yeah, so yeah, that's a great question. So what's happening is that in the POPs framework, it's basically a linear regression model where they have the magma score, magma gene level Z scores as the Y, and then these the features are based on the neighbors of every gene in the PPI network, annotated as 1 and others as 0 and things like that. So everything that in the POPs framework, everything goes through this linear regression model, with some of the features being driven by the PPI, some of them driven by the pathway data. On the other hand, for the polynet, we can actually get a better sense of the overall complex interactions that are happening between the different genes, and we do not need it to be kind of linearly related to a particular score.
So yeah, I will not bore you again.
No,
the last question is
Yeah.
that do you really think that because I was very intrigued to see that data that Alzheimer's disease has a tendency with monocyte or any functional implication, do you think or have you worked on for future plan?
Yeah, so I think one of the things that we are interested in is to, and one of the reasons why we are, of course, we looked at some of the genes that are constituting these cell types for monocytes and the dendritic cells. And one thing that I can mention, which I do not show here, is that if we look at brain-related cell types and linked to Alzheimer's, then one cell type that gets picked up is this microglia cell type, which has a very strong association with Alzheimer's and has been shown in several recent papers as well. And very interestingly, the top gene that we prioritize for microglia Alzheimer's association is this ApoE4 gene, which is also a very well-known gene. So the idea here is that once we identify this cell type and we identify what are the genes that are prioritized in this particular cell types, we can do some functional follow-up experiments, in the sense that, let's say that I am interested in microglia with Alzheimer's connection. So now one nice thing about this approach is that it tells you what are the enhancers for these particular genes as well. So now you can do like a CRISPR-I on targeting the enhancers to do like knocking down those particular genes, and you can do that in the iPSC cell lines, but you can make them differentiate in a way so that it develops into microglia. So now you know basically that, okay, if I am knocking down this particular gene, which cell type I should be targeting to kind of to provide the cues for this particular iPSCs to differentiate into, and then maybe see the function of this particular knockdown in that particular cell type. So that's the information that we can get from this particular aspect.
So I have snippet gene A, and this try checking how many times that this gene is interacting with other magma genes. Am I correct?
Yeah, so basically the magma genes are implicated by the fact that they have a high density of GWAS signal around that gene. And then on the polynet method, we are basically seeing that on the string PPI network or on the co-express DB network, how much the other genes are interacting with the magma genes.
So I have split into different genes, but those proteins actually form a large polymer. So they form a large, suppose a sample kind of thing. So typically they would, how would these polynets would rank them? Because they are interacting in a similar manner with similar genes, similar proteins. So how would they?
Yeah, so basically the protein-protein interaction, if I'm hoping that might take into account the fact that these two proteins from these two genes are interacting. And so now if it so happens that both of these genes are implicated by magma, because you're saying that both have GWAS signals. So what will happen is that you will have both of them as nodes, and then when you start from the one and then the traversal kind of goes through that node as well as the other node that you're interacting. So then both of them will get even a higher prioritization.
So they will, so polynet will rank them almost.
Yes, yes. Polynet will rank them equally, and also Polynet will probably rank them higher than a gene that is only implicated by magma and that is not interacting with other magma genes.
you say that somehow one of your goals is the predictability of this PRS?
Yeah.
But as you mentioned, and the major is the population stratification. Not quite clear that how this method would actually help in that aspect.
Yes, yes, that's a great question. So, okay, so one of the things that we are particularly, one of the research directions that we are particularly interested in when it comes to the PRS is the transferability of the PRS models from one population to the other population. So for example, if you have a PRS that is based on European data and you are applying it on African population, then the predictive power is really low. Now, one of the things that we have been able to show, and this is Westbroad et al. 2021 paper from our group, has shown that you can first perform a fine mapping of the causal variance in the European data, and you can improve the fine mapping aspect by using a lot of the functional genomic datasets. And if you can prioritize, if you can identify the causal SNPs really well, you can actually improve the PRS that is, so the transferability of a PRS based only on the causal SNPs is going to be higher. So because one of the things that mainly causes the problem with transferability is the differences in the LD that you see between different populations. But if you can identify what the causal variant is, then you can actually use that information to improve your transferability of the PRS score from one population to the other population. And we do see a drastic increase in the overall predictive power of a European PRS in the African populations if that PRS is functionally informed. And now one of the things that we can do here is that we are basically generating a lot of base pair resolution functional data from these different tissues and cell types using this CV2F. And we are now using both the genetic data as well as genomic data to kind of better inform what are the functional variants, because these are base pair resolution functional genomic data. So if we can do that, then the hope is that we can actually use this to improve this transferability even further compared to how it is currently based on just a set of annotations that are not base pair resolution.
This not final, but there can be different other steps.
Yes, that we will. So that we will not be able to account for. We will only be able to account for the differences that is based on LD. So there is definitely an upper limit to how much we can improve the transferability. One of the things that we can do when it comes to the causal SNPs being different for different populations is probably about collecting more functional genomic data from ancestrally diverse populations. And one of these consortia, for example, this IGVF consortia is kind of mainly trying to do that, where they are planning to generate a lot of the QTL data sets for different populations as well. So that is something that we would need, that kind of data to identify like a CV2F score for Europeans or CV2F score for Africans based on, also we can use the GWAS. The UK Biobank GWAS data that we are using currently for training the CV2F is also kind of biased towards white British individuals. And maybe we can generate a newer CV2F scores targeted more towards these different ancestries if we have that level of information, which probably we will be able to get there in a couple of years probably. Yes.
Two quick questions. One is CV2F.
Yes.
I've heard that, I may have heard this wrong, that we are using from multiple cell type, multiple cell lines in order to construct that CV2F. Why would you do that? Because it might cancel out, right? The effects might cancel out because one particular gene may be very highly expressed in one cell line and it may not. So if you pool these, actually the effects might cancel out. So when you're computing delta F, you might actually have a very high delta F in one cell line, and if you pool fully wash out those effects.
Yes, that's a great point. So, I mean, one of the things that, because in our this, so in the CV2F model right now, it is like a GWAS. We are basically using all the variant level features across different cell types, and as you mentioned, that there can be some effects that can just because we are pulling so many different cell types together, effects can get diluted or get cancelled out. And that's why I think doing this operation at the level of a cell type or tissue specific CV2F is even more important, because, and that's probably what we want to do, like just use a lot of the tissue and cell type specific or cell line specific data sets that we have at our disposal and probably generate one for this particular cell line or this particular tissue. And that's actually the plan going forward. One of the immediate plans right now is to do this for GM12878 and for K562, for which we actually have a lot of this allelic imbalance and experimental and deep learning data available, and then use that to create a combined V2F for those cell lines. The problem with other cell types is that we do not have that much of genomic data for other cell lines or for other tissues. So, and one of the restrictions also is that here we are just using a general set of traits and looking at fine mapped variance in a general set of traits versus non-fine mapped. Now, when we want to go into some particular cell types, if we have to keep using this same framework, then we need a trait that is related to K562 or related to G, and that is possible. Like for K562, we can use the RBC related traits, or for GM12878, I can use like WBC count or some related traits. But even for brain or blood, we can do. But then for many other cell types, it becomes kind of harder to choose like what set of traits I should be focusing our analysis on. But of course, I mean, at the level of tissue, I think it probably would be a little easier with like maybe for pancreas, we can use type 2 diabetes and things like that. But so this was like probably the first step of like many CV2Fs that we are trying to generate at tissue and cell type level. But we just wanted to see that whether at a broader level it is giving us something that is functionally important and useful from the point of view of performing follow-up experiments on.
Cognizant of the limitations of PRS at this stage,
yeah,
if you compute the correlation between polynet and PRS in a European population, let's say, what kind of correlation do you get?
You mean polynet? So polynet is kind of gene prioritization score.
But you could use that also to predict, right?
Oh, okay.
Or cardiovascular, let's say, you could also use that to predict.
Yeah.
So essentially, the prediction score that you get from your polynet and the PRS, which is again a prediction score essentially, what would be the relationship?
Yeah, so I mean, we haven't looked at a PRS that is based on like the SNPs that are linked to the genes in the polygene score. So, I mean, that is something kind of worth trying. I mean, one of the things that, I mean, the reason why we did not look at the polygene scored PRS is because polygene also uses like the GWAS data itself. And so if we, I mean, we can probably try to use the European GWAS data for building the polygene, but maybe see whether in African data we see some GWAS hits in the same genes. But it is a little complicated because I mean because I mean from the point of view of the overall PRS, maybe what we need to do is look at variants that are linked to genes that are implicated by polygene, and then maybe look at the transferability of that PRS in other populations, because we are building the polygene using the GWAS data itself. So it might be an overfitting issue if we use it on the same population.
I'm not thinking about transferability. I'm thinking about these two scores, both predictive, how well do they predict, I mean, in terms of correlation between the scores. I mean, even if it is on, I understand the problem of overfitting because you are using GWAS to construct the score and then, but even so, I was just curious.
Yeah, I think like one of the things that could be, I believe that compared to the polygene, maybe if we use the CV2F on top of the polygene, that may be, better, because the polygene is mainly focusing on genes that have that are either GWAS implicated by GWAS, or maybe that are interacting with genes that are kind of GWAS based. But on the other hand, for the CV2F, we are looking at like a bigger spectrum of variation, where the causal GWAS variance can lie, and a lot more about the distal regulation component can also be captured by the CV2F scores, which the polygene score will miss.
I have no question. There's a piece of information I want. Identify that tag. Can you suggest some good?
Like good papers. Yeah, like I mean, to identify the tags in the sense like the enhancer gene interactions, you mean? So, okay, so there is this paper, so there is Fulco et al. 2019 Nature Genetics paper is there, and then there is also Nasser et al. 2021 Nature. So those are the two references for this activity by contact method. I can send if you I can send you the links to those papers as well. The roadmap one is basically from the roadmap paper. There is a 2017 paper in genome biology where they looked at this roadmap enhancer gene linking. Sorry. So basically, so here what we are doing is just looking at the union of those two enhancer gene predictions from those two papers. But mainly we are using the one from Nasser et al. 2021 Nature. Jesse Engris is the last author of that paper, and the roadmap enhancer gene is Liu et al. 2017 Genome Biology.
All right. So thank you, Kushal, for a wonderful
Thanks.
talk. There's another. Oh, me.
For all those who are staying back for the research presentations. Okay, come back. Meanwhile, we'll start. So we have two speakers today. Our first speaker is Dr. Kushal K, and he is working as a project manager in Genome India. Dr. Payne is going to be talking about his work on SARS-CoV-2.